All right, let's dive right into this explainer. Today, we're putting on our graduate-level lenses to examine the incredibly sophisticated molecular mechanics of viruses and prions. We're going to zoom way in on the biochemical level to see exactly how these microscopic agents execute the ultimate cellular heist. If you want to understand the true molecular foundation of virology, you are absolutely in the right place.
Here is our syllabus for today. 1. A kind of borrowed life. 2. Bacteriophages and CRISPR defenses. 3. The retrovirus replicative cycle. 4. Mechanisms of viral evolution. And 5. Prions, infectious misfolded proteins.
Section 1. A kind of borrowed life. We're looking at the biological gray area between life and chemicals.
To really wrap our heads around this gray area, we got to remember viruses are obligate intracellular parasites. Basically, outside a host, they are metabolically completely inert. And the scale we are dealing with here is wild. A typical virus is a tiny, 20 nanometers across. But then you have these massive discoveries like Pithovirus siberica measuring an incredible 1,500 nanometers. It packs hundreds of genes, even ones for cellular functions like translation and DNA repair. This literally blurs the evolutionary lines between complex chemicals and actual life. Like, did they predate cells, or are they just runaway cellular fragments?
Now, zooming back down to that standard nanometer scale, a virus is essentially a masterpiece of structural efficiency. At its core, it's just a nucleic acid payload, which could be double or single-stranded DNA or RNA wrapped in a protective shell called a capsid. And this shell is built from repetitive protein subunits known as capsomeres. Whether they're forming rigid helical rods or complex, multi-sided geometries, these capsomeres thermodynamically self-assemble right around the genome. It's brilliant because it requires almost zero energetic input from the virus to build these highly stable, infectious structures.
Moving on to Section 2, bacteriophages and CRISPR defenses. Let's look at the intense molecular arms race happening inside bacteria.
So double-stranded DNA phages basically wage war on bacterial hosts using two totally different but equally fascinating strategies. First, you have the virulent lytic cycle. This is a rapid, destructive takeover, where the virus actively shreds the host DNA, builds an army of clones, and literally bursts the cell open. On the flip side, we have the temperate lysogenic cycle, which is an absolute masterclass in stealth. Here, the viral DNA circularizes and seamlessly integrates into the bacterial chromosome, essentially hiding out and passively replicating every single time the bacterium divides.
Now, when the viral DNA is in this integrated state, we call it a prophage. A single repressor protein keeps almost all the viral genes completely silent, guaranteeing the virus's genetic survival across countless bacterial generations. But here is the crazy plot twist. Sometimes the handful of prophage genes that actually do get expressed completely change the host's phenotype. For example, the bacteria that cause diphtheria, botulism, and scarlet fever, they're usually pretty harmless. They only become dangerous after they fall victim to a viral infection themselves, acquiring a prophage that specifically codes for those deadly toxins.
But, bacteria don't just take this lying down, they fight back. When a bacterium survives a phage attack, it captures pieces of that fragmented viral DNA and integrates them between repeating palindromic sequences in its own genome. If that exact same phage ever attacks again, the cell transcribes this region. This RNA acts like a highly specific homing beacon, a guide RNA, that directs a Cas nucleus to find and specifically slice up the complementary invading genome. It is a ridiculously precise adaptive molecular memory bank.
Okay, Section 3, the retrovirus replicative cycle. It's time to deconstruct the HIV molecular heist.
Animal RNA viruses are categorized by how their genomes function. Class 4 genomes can immediately act as mRNA, and Class 5 serve as a template. But Class 6 retroviruses, like HIV, they are in a league of their own. They literally violate the central dogma of molecular biology by completely reversing the standard flow of genetic information.
Because normal host cells have absolutely no machinery to read RNA and write DNA, retroviruses actually have to pack their own enzyme, reverse transcriptase. Once it's dropped into the host cytoplasm, it forces the synthesis of a DNA strand directly from the viral RNA template, ultimately creating double-stranded viral DNA. And here's an absolutely crucial detail. Reverse transcriptase is incredibly sloppy. It completely lacks proofreading ability, meaning it constantly introduces mutations. This is a massive advantage for the virus when it comes to long-term immune evasion.
Next, this newly minted DNA gets shipped into the nucleus and permanently embeds itself into the host's chromosomal DNA, becoming what's called a provirus. Now, remember the bacterial prophage we just talked about? That can pop back out,
An animal provirus doesn't do that. It never leaves. It effectively tricks the host's own RNA polymerase into transcribing viral genes just as if they were its own.
Let's put this all together and watch how HIV completely commandeers the cellular factory. It starts with highly specific binding to the host cell's CD4 receptors, which kicks off that reverse transcription process we just covered. Once the provirus integrates into the nucleus, the host cell unwittingly does all the heavy lifting. It translates viral mRNA into capsid proteins right in the cytosol and pumps out viral envelope glycoproteins over in the ER and Golgi. Finally, the new viruses bud out, literally cloaking themselves in the host's own plasma membrane so they can sneak away undetected.
All right, Section 4, Mechanisms of Viral Evolution. Let's talk about the origin of emerging viruses. Given all this intricate, highly tuned biology, you might be wondering how previously rare viruses suddenly explode into massive global epidemics. Well, emerging viruses really rely on three main drivers. First, remember that incredibly error-prone reverse transcriptase? That high mutation rate constantly churns out relentless new genetic strains. Second, modern global travel means a virus can jump from a small, isolated population to the other side of the world in hours. And third, these newly mutated viruses frequently jump the species barrier, moving from natural animal reservoirs straight into humans. If we look at the history of influenza, we see a pretty terrifying pattern of this animal-to-human transmission. Those letters H and N, they refer to vital viral surface glycoproteins. Now imagine an animal host, like a pig, gets infected with multiple flu strains at once, say an avian flu and a human flu. Their segmented RNA genomes can literally mix and match during viral assembly inside the cell. This triggers what's called an antigenic shift. It's a massive genetic reassortment that creates completely novel strains, unleashing devastating pandemic waves on human populations that have absolutely zero pre-existing immunity.
And finally, Section 5, Prions, Infectious Misfolded Proteins. We are looking at pathogens without nucleic acids. If you thought viruses existed in a gray area, our final topic actively shatters the established laws of infectious disease entirely. We have to ask, how on earth can a protein, something completely lacking nucleic acid, act as a transmissible pathogen? This exact question deeply puzzled scientists who were studying horrific neurodegenerative diseases. For decades, the golden rule of biology was that infectious agents had to carry genetic material to replicate. So finding an infectious agent made entirely of protein, that violated everything we thought we knew about genetics and inheritance.
The answer lies in a devastating molecular chain reaction. Basically, a prion is a misfolded protein that physically forces normal proteins to undergo a radical conformational change. They often shift from functional, soluble alpha helices into these highly stable, abnormal beta-pleated sheets. When a single prion enters a cell, it bumps into normal proteins and converts them. Then, those new prions clump together into practically indestructible amyloid plaques, converting even more proteins in this endless loop. It is an incredibly slow but universally fatal accumulation in brain tissue.
Throughout this explainer, we've dug into the astonishing mechanics of genetic heists, reverse transcription, and runaway protein chain reactions. As we untangle these devastating molecular events at this graduate level, it really forces us to step back and ask, are viruses and prions nature's most sophisticated, complex biochemical associations, or are they actually the absolute simplest forms of biological life? It's a wild thought to chew on. Keep exploring, and I'll see you in the next explainer.